Phospholipids or the membrane lipids are the major constituent of cell membrane. Like fats, they are composed of fatty acid side chains and a phosphoryl head. Specifically, there are two fatty acid tails attached to a phosphor group to form a polar head. The phospholipid is an amphipathic molecule, meaning it has hydrophobic part and a hydrophilic part. The fatty acid chains are hydrophobic and cannot interact with water, whereas the phosphate containing head groups are generally hydrophilic and interact with water. The negative phosphoryl heads are composed of phosphatidylate. They can be either phosphatidylglycerol or cardiolipin. Cardiolipin is a kind of diphosphatidylglycerol lipid because it has two phosphatidic acid moieties connected with this glycerol backbone in the center to form a dimeric structure. So it has four distinct alkyl chains and potentially carries two negative charges. As there are four distinct chains in cardiolipin, the potential for complexity of this molecule species is enormous. Negative phosphoryl heads are generally phosphatidylethanol amine and have a net positive charge. They are involved in interaction with DNA and transporter proteins. Saturated fatty acids, which are generally 6 to 22 carbon in length, with straight tails are compressed by decreasing temperature, and they will press in on each other, making a dense, fairly rigid membrane. Some of the bacteria have unsaturated fatty acids. These unsaturated fatty acids are compressed and result in the king tail elbow adjacent phospholipid molecules away, making more space between the phospholipid molecule. This elbow room helps to maintain fluidity at temperatures which membrane with high concentration of saturated fatty acid tail would freeze or solidify. As a result, they help the bacteria to survive under extreme low temperatures. Because a cold environment tends to compress membrane composed largely of saturated fatty acid, making them less fluid and more susceptible to rupturing. Similarly, cyclopropane fatty acids are widespread in bacteria. As their exposure of hostile environment, the concentration of cyclopropane fatty acid increases to protect the membrane.
Plasma membrane, also called a cytoplasmic membrane, is present just beneath the cell wall. Several models were proposed to explain the structure of plasma membrane. However, the most widely accepted theory is fluid mosaic model, which was proposed by Singer and Nicholson in 1972. According to this model, the plasma membrane is formed by two layers of phospholipids. These two layers are arranged opposite to each other, with slight overlap. The plasma membrane is mainly composed of three components: phospholipids, protein, and cholesterol. The phospholipid molecule is composed of a phosphate group and a lipid. Phospholipid is an asymmetric molecule with a polar end and non-polar ends. The polar end of phospholipid is hydrophilic, and the non-polar end is hydrophobic. The two phospholipid layers are arranged in such a way that the polar ends of phospholipids of each layer are directly exposed to internal and external environments of the cell. The reason for this arrangement is, the internal and external environments of the cell are aqueous solutions in many cases. And the polar end being hydrophilic, it always interacts with water. And, The nonpolar ends are left free inside. This is the reason why, in the nonpolar solvent, this condition may get reversed. And as a result, the membrane gets damaged. The phospholipid bilayer is integrated with protein molecules, distributed throughout the layer. Some proteins are bound to the external part of the layer. These are called as peripheral proteins. Some proteins are totally merged into the bilayer, and are called integral proteins. The integral proteins are so strongly bound, that they can only be separated when the membrane is destructed. The peripheral proteins are loosely attached, that they can be relocated their position within the membrane. The cholesterol molecules present in the membrane provide stability to the bilayer. They help the bilayer not to get separated too much in different environment conditions. The main function of plasma membrane is to control the entry and exit of selective biomolecules from the cell, and also protects the cell from its external environment.
Openouts are pentacyclic hydrocarbon derivatives of sterol and function similar to that of cholesterol in eukaryotic plasma membrane. These molecules tend to dampen the effect of temperature on the membrane. Thus, it functions as fluidity buffer, preventing low temperatures from inhibiting fluidity and preventing increased temperature from increasing fluidity too much. One of the major differences between archaea and bacteria is the composition of the cell membrane. So unlike eukaryotes and bacteria, archaeal membranes are not made up of fatty acid attached to glycerol backbone. Instead, the polar lipids consist of isoprenoid molecule, which is about 20 to 40 carbons in length. These chains are usually saturated and either bounded to glycerol by ether bond. They have L-glycerol, tropanoids, and sometimes, in some cases, they also have a monolayer instead of a bilayer, which is formed by tetraether-linked lipid transmembrane phospholipids. And they also lack unsaturated lipids and have cyclopentane ring. As you can see a comparison of the fatty acid side chain and phosphohead. As you can see in bacteria and eukaryotes, there is an ester bond, and in case of archaea, it's an ether bond between the fatty acid side chain and the glycerol. And the fatty acid side chain, as in case of bacteria, is not branched, whereas in case of archaea, it's branched, called phytenyl side chain. You can see the ether link between the glycerol and the phytenyl side chain, and you can see there are two bonds with the glycerol here, and in the second case you can see the formation of a lipid monolayer in which the glycerol from one leaf is attached to the glycerol from the another leaf by a single biphenyl chain. So here you can see a lipid bilayer. Also, when phenyl chain is attached to each other from to the glycerol from the opposite sides, it form a lipid monolayer. This is called diglycerol tetraether bond. And lastly, they have cyclopentane ring, as you can see here, which change the fluidity of the membrane. The key points of today's lecture are: the prokaryotes are divided in two groups, that is, eubacteria and archaebacteria. Prokaryotes vary greatly in their size, shape, and arrangement, which helps in identification of bacteria. The cell membrane of bacteria have proteins which play a vital role in signaling, and the cell membrane of archaea is quite different from that of eubacteria and help them survive under extreme environmental conditions.